Good afternoon. Today we are talking about design and development of vaccines. Our group is composed by Magda Garcia, Ingrid Valentina Reyes, and Nicolas Torres.
Okay, to start, from our profession as pharmaceutical chemists, our objective is to make our college understand what the process is like to carry out the development of a vaccine, starting with the previous concepts related to this topic and later emphasizing in each one of the development phases. To start, it's important to define some previous concepts that will help you to understand what will be explained later. First, what is a vaccine? This is the first concept and in fact the most important to understand this topic. A vaccine is a preparation that is administered to generate active and lasting immunity against a disease by stimulating the production of defense, that is antibodies. Antibodies are proteins generated by our body to induce a defense response when it detects harmful substance called antigens. The next concept is immunogenicity. This refers to the ability of a substance to generate immune response, that is when the body responds to foreign agents. Vaccines produce immunogenicity, that is a desired event. In other words, the aim is that the vaccine has the capacity to generate an immune response so that the body defends itself against viruses or bacteria that generate a specific disease. In order to promote the available free circulation and use of a vaccine, it's necessary that they maintain a balance between efficacy and safety. But what does it mean, these concepts? Safety, in terms of a vaccine, is the property of this vaccine to produce minimal adverse effects, as well as not generate serious adverse effects. Efficacy, this refers to the ability of the vaccine to significantly reduce the incidence of disease or infection.
Well, the parts of a vaccine. The first one, we have the antigen. It is the active compound of a vaccine. This is a modified or partial form of the virus, bacteria, or the toxin that causes the disease against which the vaccine protects. The vaccine antigen is no longer cause disease, but it can produce an immune response. The second one is the adjuvant. The substance used to enhance the immune response to a vaccine. One way that the adjuvants improve the immune response is by keeping the antigen near the injection site so that they can be easier accessed by cells of the immune system. The third one is the preservatives. They are used when you want to prevent fungal and or bacterial contamination of vaccines because these products has to be sterile. The fourth one is stabilizers. These substances are used for help to keep the antigen and other components of the vaccines adhering to the size of the vaccine pill. Well, we can put this part in an example. Imagine a car when you have the antigen is a part of the virus inactive or a substance that causes the disease. He is the driver. We have the adjuvant that keep the car in the correct way and guide the antigen to the task. The preservative maintain the car clean, avoid that invasor enter into the car, and finally the stabilizer keeps the odor inside the car and all the components united.
Design and development stage has three important stages. First, basic research, clinical research, and post-authorization that we are going to explain. First step, basic research. The initial question begins with the specific need or expectation about one problem that you need to figure it out then. Research, research have to carry out principal problem, propose hypotheses, and fix aims investigations. For example, in this moment the world need a medicine that treat COVID-19, and the principal question could be, if research developing a vaccine, it will avoid people get sick and die. Then research must identify the antigen, and it is one of the most important and difficult steps during investigation. They must identify the structure of the virus and its composition, chemical and physical factors that are active or inactive the virus. The next step will have to perform animal tests for evaluating toxicity, damage in DNA like cancer, tolerance, and the most important, will have checked if it will have generate an immunitary answer. After all tests had carried out and research figured out some prototypes that could function, they must begin the investigation with humans. Research have to ask for legal permit to test in humans.
Now, when a new vaccine has government authorization for research in humans, the clinical study begins through the research phases presented below. Clinical Research Phase 1. In this phase, the studies are carried out mainly in a small group of healthy volunteers, approximately between 20 and 50 volunteers, who will be closely monitored. The main objective of this phase is the preliminary evaluation of the safety and immunogenicity of the vaccine. First, safety, which is an indispensable factor, is evaluated. This evaluation is carried out by observing tolerance at different doses, monitoring immediate toxic effects, and monitoring clinical effects such as fever, local swelling, among others. After evaluating safety, a detailed immunological study is performed to determine if the vaccine is effective.
Well, the clinical research phase 2. This is safe in the clinical phase when you pay attention in the security, tolerance, and dosage range. The dosage range is an allowed level of the antigen that can be able to generate an immune response instead to generate an infectious process. The difference between this phase and the other is the number of people implicated in the test. The people is between 200 and 500, and in this phase you start administering the vaccines to healthy group and the sick group, and these tests are the beginning to evaluate the effectiveness of the vaccine. In a sense, this phase is for evaluate the activity of the vaccines against to the infection disease.
Clinical Research Phase 3. In this phase, increase the number of the patients who takes the vaccine. For example, the number could be above 1,000 participants. In this line, the experiments will perform in multiple hospitals with controlled and random clinical trials. In this phase, research will have evaluating immunogenicity, safe and efficacy, and the most important is careful analysis on all this information in a lot of patients that they have been guarded. Finally, research have to ask for legal permits to making that these vaccines have an organization and commercial conditions for its sale around the world. Third, post authorizations and commercialization. Post authorization and marketing study have been like aim vigilance and control of the medicine, complete characteristics and effects about vaccine activity in all types of race around the world. In this line, this phase allows authorization extension in patients with a specific disease and identify possible risk factors or effect modifiers in specific patients. The federal agencies are responsible for authorizing the medicine in each country and control and supervision of the vaccine effects.
Finally, we have the conclusions. Vaccines are developed, tested, and regulated in much the same way as other medication. In general, vaccines have more meticulous testing and involve more human in vaccine clinical trials. In addition, vaccine monitoring after official authorization is granted. Is carefully evaluated by the Centers for Disease Control and Drug regulatory agencies like FDA and EMA. This development of a vaccine is a long and complex process. This process can take from 1 to 2 years until 10 and 15 years and involves the combined participation of public and private organizations. Well, we are finished our presentation. Thank you for take the time to see our presentation and goodbye.